All right. Let's talk a little bit about the next lab. If you go into the course in Schoology, you are going to be seeing, I've changed the landing page. I don't know how many of you have noticed this. Basically, I've changed it to updates. I still think messages are the best way to communicate with me directly because I get more alerts from the messages, but this updates page will be course specific updates that I want everybody to see when they first click onto it. And I've been convinced by other teachers that this is a better model to use. If you disagree and you want to say, Mr. Burns, go back to messages, let me know that. I mean, send me a message and we'll say, I need messages. You have to click on materials on the left-hand side now to see what was our old landing page, our materials page. And if you go into the Flex Learning folder, a couple of things that I'd just like to highlight. I am going to be updating today the closure update form in probably this sometime between 11 and noon with the updates from the AP board on your exam. I will be, the textbook access has been added for those of you who don't have access to the textbook. So if you go into Unit 3.1, you'll see a new assignment investigating the role of a new protein. Now, I've had to, because Schoology has been getting shut down, or it has shut down some of its features over the past few days. I've inserted this not as a Google assignment because that's not supported well by Schoology, but rather just as a link to a Google Doc that when you open it is going to prompt you to make a copy. So you'll need to make your own copy and file this somewhere where you can find it. Probably in a folder titled enzymes or something like that, or topic 3. So you'll have your own local copy. That's what you're ultimately going to submit back to me for grading. But this will also allow you to edit it. But when you open that, it is going to look like this. It's going to be called Investigating the Role of a New Protein, and there'll be some video instructions, which are the ones I'm giving you right now. Let me click back into the meeting here. Any questions? I don't see any yet. All right. If a question comes up and I'm not noticing it because on my screen I can't see the chat and the page I'm working on, just click in and just say, I have a question. So otherwise I'll click back to the chat every once in a while. There's some background here, which is the sort of reason for this experiment. The background is basically this. We discover new proteins as scientists all the time. But when you look at a protein, it has a shape, but the shape itself doesn't always tell us what it does. We have to kind of backwards engineer the role of a protein, either by figuring out where the protein's found and what kinds of substrates are present in that environment, or which is what we're going to do here, by actually isolating that enzyme and then testing it in a variety of situations to see what it does. The location of where we find a new enzyme or a new protein can tell us about the environmental conditions that are optimal for the enzyme activity, because evolution will affect the shape and structure of that enzyme. But it's not always obvious what the function of a protein is. So we know, oh, that protein's probably going to be most effective at around, 22 degrees Celsius, or it's going to prefer this kind of pH, but we won't know really what is it doing there. So this lab is supposed to model that. So you're going to open this website that we have been using. And you're going to see this simulation here. That is different than the last one we worked on. So, but it simulates work with enzymes, and we have variables here. Here's those environmental variables and substrates. But you're going to click on the chemical indicators in the top. And it's going to set up a similar page. Again, we can control substrates and enzymes and environmental variables. We have some graphs here, and then we have these things called indicator solutions. And indicator solutions tell you about the presence of certain products or reactants. And so in this test tube, we have an indicator called Biuret, which indicates whether there is protein or not. In this case, it's purple, so we see that there is protein in this sample. Now, if those proteins were disassociated into their building blocks, which hopefully you know are amino acids, then we would understand that the proteins were no longer present and the solution would change color. There are questions about that. Okay, when we make a copy, Lindy, I'm reading your question right now. When we make a copy of the lactase temperature. I'm not sure, Lindy, that I understand your question. When we make a copy of the lactase temperature lab, the graph. I see. You don't actually, the lactase temperature one was mine. You don't need to make a copy of that. You don't need to do anything for that one. That one is really just to look at to see how to do it. I probably shouldn't have put that in there as an assignment because I just sort of did that one with you. So I think I'm answering your question there. The one I'm going to be assessing and the one I really want you to be doing is whichever one you were assigned to do, whether you were assigned to do pH or substrate concentration. Got it. Okay. So we've got this simulation up. And we have a bunch of different things we can control. So in the investigation, you're going to be assigned to a specific enzyme to investigate. And if you open this document link here, you're going to get a table. Your name will be in it as well as an enzyme. Okay. And you're going to put that enzyme in here as number 1. So let's say your enzyme is amylase, right? Right. So you're going to select that enzyme. That's the only enzyme you need to test. You can ignore the other enzymes unless you just want to play around with them. And then you need to recognize that amylase is going to be put into a solution that is set at certain environmental variables. So before you continue, well, you kind of have 2 choices here. You can either mess around with the environmental variables to see what happens. Or you could go and do just a Google search of amylase. So you could look up where amylase is found in the body, and that should give you a clue about the ideal temperature and pH situation. So if it says, oh, it's found in the stomach, then you would maybe research, well, what's the pH of the stomach? And that would probably be a good starting pH to use. If you find out that it's present in the mouth, you might look up what the pH of the mouth is or the temperature in your mouth. And those would be good values to enter in for your environmental variables. Okay. You'll have your enzyme selected, and then you'll have a bunch of different substrates or things that are in the test tubes, right? So you have a test tube that has nucleotides, right, which would be combined to form DNA or RNA. You know, you have proteins, you have lipids, you have starches. So one of the things you're going to need to do is pay attention to, are these the monomers or the building blocks, or are they the polypeptides, or not polypeptides, the polymers, the fully constructed molecule? And then you need to think about, like, are they going to be broken down by an enzyme, or will they be built up? Okay. If you run this, you're going to get a graph that's going to show something happening, right? So if I run that again, or if I just reset it, I'm going to get some data here. It's going to show me the indicators. So this one has no starch. This one has starch. This one has proteins. And this one says there's no DNA in it, right? So if I run it, what did I do? Whoops, I took the amylase out. When I reset it, the amylase changed. So anytime you click reset, you're going to need to make sure that you re-put in your values. Okay, so now of those, only one of them changed. right? There are now disaccharides present over time, and my indicator has changed to go from having starch to no starch, which means something happened to the starch. Okay, you may need to go back to your try one work on macromolecules and just kind of learning about the substrates that or the monomers that build different polymers to interpret your results. Okay, so once you've done this, you are going to basically play around until you feel like you have an understanding of how to prove what your enzyme does. So in question 2, you're asked to identify the variables, the environmental variables you set up to run your experiment. Again, play with those a little bit to find an optimal value and then insert them. Go do some background research. When you do that, you're probably going to find out, oh, amylase, it's an enzyme. It breaks down starch into glucose, right? So when you do that, you might be tempted to say, well, that's my claim. And it can be your claim. You're going to find the answer to what these enzymes do through a Google search because these are well-researched enzymes. But I'm asking you to try to model what would happen if we didn't know the role of the enzyme. And as biologists, that's where the new interesting research is, right? Is looking at all the thousands and thousands of enzymes that we haven't identified yet and figuring out what they do, right? And so I want you to imagine a scenario where you're trying to figure out how you would do that. So, whoa. That was weird. I don't know if you guys saw that or not. Okay, so no questions right now. And so your task essentially is to create a claim about what the enzyme does. So I would write a statement here if I was amylase about what amylase does. And then I need evidence that supports my claim. And the trick here, if you want to earn full credit on this, is that you not only need to prove what your enzyme does, but you also need to prove that it doesn't do other things, right? So let's say you're on this and you say, oh, well, this graph indicates that it does something to starch, and you're going to write something about that. And you want to use this data as evidence. You might want to have this as your evidence. The best way to do that is to do a screen grab. And if you guys don't know how to do this, you're going to click on Control Shift, and then along the top keypad, there's one that looks like a little square with 2 lines next to it. If you click that, it's going to gray out your screen, and you can highlight, let's say you wanted this graph, right? If you copy that to the clipboard, right, you can come in here and you can insert that graph. Now, anytime you insert a graph, you need to give it a name. So we call this figure 1. And this would be a description, production of disaccharides by amylase, I'd write about what this figure shows. And then over here is going to be your reasoning, okay? And in your reasoning, you're going to explain, why does this evidence support the claim you made, right? And so you might say, well, disaccharides are the monomer of starch, and so starch molecules must be broken down into their monomers by the enzyme carrying out the reaction. You're going to write that out. Now, but you're not done when you do that on its own, because that only proves that amylase breaks down starch, for instance. What it doesn't prove is that it's not involved in other chemical reactions. So you're also going to need to prove that, okay? Now, when you set up your initial variables, right? You're going to need to tell me what your environmental situation is and what your test groups are, right? So what's present in the test group when you run it. If you're going to have no lipids present or no starch molecules present or no proteins present or no whatever, you should indicate what was in the solution and how the results help you know that. And what I'm going to be looking for is, did the experiment you set up actually demonstrate conclusively the role of that protein? And added to that, does it demonstrate that the protein's not doing, or the enzyme isn't doing something else? Okay. So hopefully that gives you guys enough understanding of what to do going forward on the enzyme work. That's going to be due on the Tuesday after spring break. So I'm going to stop presenting here. Come back to my home screen. Make sure I don't have any questions. Okay, so are there any questions on that? Okay. Well, so let's talk about some of the other things. The AP board just came out with some additional information, which I'm going to put in our update folder or update file about the testing dates and times. I think, pretty sure that your test date is May 18th. I should have written it down. It'll be on that updated form when I do it. But I think it's on May 18th, and it's going to be at 1 o'clock in the afternoon. And everybody's going to have to take, for us here in Central Time, everybody's going to need to log on and take it at the same time. There's going to be, before that date, plenty of opportunities to practice, making sure you get logged on. You'll also need to have a dedicated device which you're going to log in with. And they'll put out more information about how that's going to work as we go forward, because they're going to essentially, you're going to be logging into a site. They're going to lock you on that screen. Now, I understand they're not going to be able to monitor the use of other devices, but you wouldn't be able to submit answers from any other devices. I think that's probably why they're focusing on FRQ response type questions, because it requires you to connect and synthesize ideas more than a multiple choice assessment does, where you can't really, you can't control whether somebody is looking up an answer, and those questions are a little bit more, individual. So let's see, why am I still presenting? I don't want to be presenting anymore. Stop presenting. Are there questions about that? Okay. So next week, I know it's spring break. You won't be needing to do any assignments for the OSSEO version of AP Biology, right? The graded class you're in right now. But you are going to want to be working on the Khan Academy work that I suggested you look at during those couple weeks while teachers were preparing their online courses. If you didn't do it during that time, this gives you another chance to go in and do that. And I will run, I guess it's going to sort of depend on interest. I'm going to put a survey. If you go into the AP Biology class and you look in our Flex Learning folder, there's a Unit 6, an optional unit for the AP test. I will be putting in some a survey here asking for kind of like what information you're interested in. No, that's not exactly right. not what information you're interested in, but like how much support you'd like. Like, do you want to do these kind of online lectures, or are these not useful? Should I just be recording them on my own? Um, you know, are there certain times that you would like to be able to meet next week? You know, like what kind of support do you need? I'm also going to post some practice FRQ questions specifically associated with Unit 6. And Unit 6 is a complicated unit, so I'm imagining that there is going to be some sort of reteaching and stuff as well, but I'm not going to assign any official like assessments that you have to do, that you have to submit, and I'm going to grade. But I will probably post some questions. I will post our learning targets in there, or learning objectives, so that you can kind of go and see whether you understand those topics. Yeah. And I will post some practice questions that you can do and then ask me about. Starting the week after spring break, we will begin doing practice FRQ assessments using old AP test questions. And I know some people were looking for resources to help them. The best place right now that I can direct you to is if you go into the course and you look. Sorry, I know you're watching me click through screens right here. Hold on, let me present my screen again. And you click on the closure updates form. Within that form, which is going to take a minute to load, there is, let's see, why is it taking so long? I have to be patient. Questions. No other questions? Okay. Come on, close your updates. All right, it's unresponsive. So if you go into the closure updates thing, there's a couple of resources in there that you can use. Those resources will AP Central has a daily lecture that they're running, I think at like 3 in the afternoon during the week, where they go review main topics from the course and do practice problems. Those are all linked. You can get to their YouTube channel and check that out there. There is Khan Academy has some review stuff that's on there. Kaplan, which is a test prep service, they're also running a free service to review for the AP test. And again, it's AP biology teachers around the country running practice problems. And then I am going to be doing the same thing. So essentially there's 3 places you could go where you could get all of the practice problems and review sessions you want to do. And the AP board feels really comfortable that with it looks like there are going to be 2 FRQ questions, they can still reasonably assess your understanding of the course as a whole. I'm a little skeptical of that. I can't imagine that is true. However, what it means is that the questions they're going to use, almost certainly you're going to get a data set figure. You're probably at least once going to have to graph and put in standard error of the means or run a chi-squared test or something like that. But you're going to get data sets and graphs. And those data sets are going to be about something that is broad enough that they could then ask a series of written response questions that would cover a variety of things. So I'm imagining like an experiment with fruit flies, where the fruit flies are, showing different traits. And then there could be a question that asks you to think about like, what evolutionary processes could relate to that? How could gene expression lead to that? How could protein structure lead to the phenotypes that create that? Or enzyme activity, or they're gonna cover a range of topics, all on the one theme of this data set about fruit flies or something like that. So I will be doing some of that work with you next week on Unit 6, and then the following week we'll begin doing that work with the other units we're covering. So that we've covered this year to prepare you for the AP test. But we also still do have some other work we have to do. We're going to finish up photosynthesis and respiration. Respiration will be next week. The next week we meet as a class, not during spring break, but and then the week after that should be respiration, and we'll be getting closer to the AP test date at that point. And so yeah, I don't know if anybody else has any questions. I appreciate you guys coming here, but I don't know if it's helpful to do this live, or if it would be easier or just as easy not. Why don't I'm going to take this off the recording right now, because I don't think we need to record me asking if you have questions.